this side of the room today. Maybe it's different view. I can't see my slides from that podium. So this is the conclusion of our receptor binding two-part lecture. Today I'm going to finish this up by talking about kinetics, and then agonist and antagonist binding, and then we'll talk about some of the applications of receptor binding. At the end of the lecture, I'm going to talk about the homework assignment, which I posted online. So I'll give you some helpful hints about that. All right, so last time we talked about radioligand binding using radiolabeled drugs to identify receptors. And one of the key assumptions that we made was that we were in equilibrium. Remember, so we did a time course, and you saw that as you increase the association of the radiolabeled drug with the receptor, there's initially a very rapid association, and then as the reaction proceeds, more and more of the drug receptor complex becomes dissociated until eventually you reach an equilibrium where the on rate is equal to the off rate. And so all of the equations and all of the discussions we had last time made the assumption that that we were in equilibrium. In fact, in the body, drugs are rarely in equilibrium, okay, because there's always things that are happening that interfere with the process of equilibrium. Concentrations of drugs are constantly changing at the target site. You have removal of the drug from the active compartment. You can have metabolism of the drug and so forth. So equilibrium is more of a theoretical thing than anything else. But today we're going to talk about the components of equilibrium, and that is association and dissociation. There is a little bit of math in here, but very little of the math that I'm going to be showing you is something that you have to be responsible for. I'll tell you exactly what it is you'll need to know. So as we learned last time, there are two components to the reactions of drugs with receptors. There's the association rate, which is defined as the association rate constant, K1, and the dissociation rate, which is defined by the dissociation rate constant, K-1. We'll begin with the dissociation rate, K-1, because that is the easiest. So this is the reaction that occurs when you're looking specifically at dissociation. The drug receptor complex, which is denoted here by D star stands for the radiolabeled drug, is bound to the receptor, so we have the drug receptor complex. And since this is a reversible reaction, once the drug receptor complex is formed, it dissociates into its free components, the rate of which reaction is defined by K-1. All right? So the larger K-1 is, the faster this drug receptor complex dissociates. Now there's the rate equation here, and I doubt that many of you remember much of your freshman chemistry, but you probably learned about first order and zero order rate equations many years ago. And you don't really have to know this very much, but here is the equation that defines this, okay? The rate of disappearance of RD, okay, so disappearance, that's where you have the minus sign, okay? So DRD is a function of time. The rate of disappearance of the drug receptor complex as a function of time is equal to K-1 times the concentration of the drug receptor complex. So this is a first order equation because it's got only one variable in it, okay? Very simple. The only variable that you've got in it is the concentration of drug receptor complex. So first order, a zero order equation means that it's linear if you plot it on an arithmetic graph. First order means that it's logarithmic if you plot it on a semi-logarithmic plot. Well, I'm sorry. A first order equation means that it's linear if you plot it on a semi-logarithmic plot. So this is the plot of dissociation on a semi-logarithmic basis. So you start out at 100% bound, and then the drug receptor complex dissociates, okay? And as you can see it go down, it looks linear here, but it's only linear because it's logarithmic on the y-axis. If you plotted this arithmetically, it would be an exponential curve, okay? It would start going down very fast and then even off at the very end. So we plot it this way normally because it's a whole lot easier to see a linear plot than it is an exponential one. But the main reason we do that is because the main thing that we're interested in dissociation is something called the half-life. The half-life of dissociation is, of course, the time it takes for the drug receptor complex to dissociate by 50%. And so the longer the half-life, the more stable the drug receptor complex is. So here on this plot, you can immediately see what the half-life is, okay? It's 100%, there's 10%, so there's 20, 30, 40, 50. There it is right there. What is that? My angle isn't very good. Maybe 3 minutes? Okay, very simple. That's all you need to do. Once you have the half-life, you're all set. Now, if you really want to be more mathematical about it, you can take a look at the slope of this line, and the slope of the line is the negative of k minus 1. So you can actually calculate a number from k minus 1 for that. But to be honest with you, most people really are more concerned about the half-life dissociation. All right, so that's really basically it. And I'm going to skip this business of how you actually define this. Normally, you can do this experiment in the test tube by simply adding an excess, a very large excess of unlabeled drug to this reaction. So once again, you form a radiolabeled drug receptor complex, you let it reach equilibrium, and then you add a very large concentration of unlabeled drug. The unlabeled drug competes with the labeled drug, but you have such a vast excess of it that once the labeled drug goes off, there's a much bigger probability that what's going to come back on again in this reversible reaction is unlabeled drug. So what you see after the point that you add the unlabeled drug, you see this decrease in radiolabel binding simply because you've added this excess of drugs. So this is a fairly easy kind of procedure, and dissociation rates and half-lives are calculated all the time. It's very simple. Association is more complicated because of the fact that it is actually a second-order equation. It involves two variables and more complex kinds of approaches. Here is the association reaction. You have the radiolabeled drug plus the free receptor. It binds to create the bound receptor radiolabeled complex with a rate that's defined by K1. That's fine, okay? But the problem is that as soon as you form the drug receptor complex, it dissociates. And of course, the rate of that dissociation depends upon K minus 1, which we've already calculated. So the rate equation for this is very complicated. The rate of appearance of the drug receptor complex as a function of time is equal to K1 times R times D. Okay, that makes sense because that's the forward reaction. But immediately, you're seeing a reduction in that amount by the dissociation. So you have minus K minus 1 times RD, which of course is the dissociation reaction. So this is what association looks like, all right? We've seen this before. I showed this to you in the last lecture. You add radiolabeled drug, immediately it starts going up very quickly, and then as time goes on, the slope of this line goes further down until eventually you reach equilibrium where the situation is in steady state. So this is a very complex kind of mathematical equation, and you can do complex math, but you can also make assumptions that make it a lot easier. You can play a little mathematical game and basically say at early time points the concentration of free drug is much, much greater than the concentration of free receptor. So part of this equation can go away, and you can create a pseudo first order kinetics. The other thing you can do is go at very early time points. So what I've done here is I've created a graph out of exactly the same data that you saw here, okay? All I've done is just look at the first 5 minutes of this reaction, all right? And you can see here that in the first 5 minutes, it's almost linear, all right? It's not quite linear. If you draw a straight line through here, you can see it's beginning to tail off here, but it's almost linear. So at early time points, okay, it is a pseudo first order reaction because the dissociation has not yet begun to take place very much. So if you really sort of restrict your time points to these early time points, you can make a pseudo first order equation, and the slope of this is something called K observed, and I'm not going to waste your time trying to resolve and derive this equation, but it turns out that the K observed is a function of the drug concentration times K1 plus the dissociation rate, K minus 1. So you can rearrange this equation and solve for K1 right here. So so the K observed is the slope. The drug concentration you already know because you know how much drug you put into this reaction. And the K minus 1 you've already solved from the previous data. So from this, you get an estimate of K1. This is just an estimate. K1 values tend to be fairly inaccurate because you're really focused in on the very early point of this time point. But it's good enough to get us close enough to figure out a little bit about what's going on. So this is the math that's associated with kinetics. These kinds of rate equations I'm not going to hold you responsible for. You don't need to know all of that. What you need to know is this. This is very simple, but it's fundamental, and it's not only just fundamental for this concept of receptor binding, but as you'll see a little bit later on in the lecture, this relationship between kinetics and affinity is of great biological and physiological importance. So there is a significant relationship between kinetics and affinity. You already know this because we define the dissociation equilibrium constant, which again, as you remember, is a measure of affinity. Remember, the higher the KD, the lower the affinity. The lower the KD, the higher the affinity. So the KD is a measure of affinity, and it is the ratio between K minus 1 and K1. Let's take a look at the units. Turns out that the units of K1 is molar to the minus 1 times seconds to the minus 1. K minus 1 units is second to the minus 1. So if you put these two things together, okay, seconds to the minus 1 on top, molar to the minus 1, and seconds to the minus 1 on the bottom, the seconds cancel out, and you end up with a unit of molar, okay? Molar. And that's exactly what we want. Remember I told you last time that we don't use the Ka because the equilibrium affinity constants, the Ka, is a direct measure of the affinity, but the units of that is molar to the minus 1, and it's very hard to work with that. That's why we use the Kd. And the Kd, the units of that is molar, or nanomolar, or micromolar, or milligrams, or pounds, or whatever you want to use, okay? Now, this very simple equation has fundamental importance in understanding the way drugs and neurotransmitters and hormones act at their receptors. Because what it means is that the ultimate affinity of the drug is determined by both its dissociation and association rates. Now, it turns out that for most drugs, K minus 1 is pretty much the same. The association of drugs at the receptor is fairly similar. Drugs don't change much according to their association rates, but their dissociation rates change a lot. And this equation basically is telling you that if a drug has a high affinity for its receptor, it will have a low dissociation rate. And if a drug has a low affinity for its receptor, it will have a very rapid dissociation rate. This is really fundamentally important because the whole system about the ways that receptors and neurotransmitters and hormones interact, and we'll see this both today and in the next lecture, involves this relationship between dissociation rate and affinity. So just remember that. If a drug has a high affinity, it has a slow dissociation rate. I'm going to give you an example of how this kind of thing works in the laboratory. Now, this is sort of a thought exercise, not something, once again, that I'm going to ask you to remember. When people are doing receptor binding assays, you have to separate the bound and the free radioactive drug. And you have a certain amount of time to do that. The longer it takes, the more dissociation of the drug receptor complex can occur. So the question, if you're designing an experiment in the laboratory, you have to determine how long you have to conduct the separation between the bound and free ligand before you reach an error that completely invalidates your experiment. So this business about the relationship between kinetics and affinity will allow you to make some judgments on this. So here is a plot which basically refers to the half-life of dissociation as a function of the KD. We've talked about this already. Lower KD values, in other words, higher affinities, have longer dissociation rates. So look at this up here. I'm going to have to angle myself here so I can see a little bit about what's going on. All right, so a normal neurotransmitter, okay, so a normal drug, I told you last time that a normal drug affinity is around nanomolar, 10 to the minus 9 molar, okay? So if we look at this plot and go over here, it looks like that the approximate half-life of a drug with a 1 nanomolar KD is 1,000 seconds. That's quite a long time. The normal neurotransmitter, okay, most of your neurotransmitters like dopamine and norepinephrine, acetylcholine, and so forth, have KDs for their receptors around 1 micromolar, okay, 10 to the minus 6 molar. Look how fast they dissociate, 1 second, okay? So neurotransmitters and hormones act very quickly, and they dissociate very quickly. That's part of the way they work. That's part of the reason that they work so quickly. This plot goes all the way down to 10 to the minus 12. I don't know too many drugs that have 10 to the minus 12 molar affinities. About the highest affinity that I've ever seen, you can create monoclonal antibodies that have almost 10 to the minus 12 molar affinity for their antigen. You can have some very, very potent interactions between antibodies and antigens. And if you have a reaction like that, what is that up there? 1,000,000 seconds? Something like that is a half-life. All right, so if you know this, then you can guess how to design your experiment in the laboratory. Let's make an estimate. How much of this error can you afford to tolerate? Let's just say that we can afford to have a 10% error, so that 10% of the radioligand bound is lost during our separation procedure. We don't want to have any more than that. Well, 10% loss turns out to be 0.15 half-lives, all right? So 50% loss is 1 half-life, all right? And 1% loss is way down here at 3 or something like that. So 10% loss, which is the most we're going to tolerate in this error, is 0.15 half-life. So all you do is to combine these two things together and say, given a certain affinity for different drugs, how much separation time do you have so that you have no more than 10% lost? Well, if you're doing your assay with a drug that has a typical affinity of 1 nanomolar, 10 to the minus 9 molar, you have 1.7 minutes to proceed with this reaction, to proceed with the separation of bound and free ligand. That's a long time in a laboratory experiment, so you're fine, you're good with that. But let's say you were doing a radioligand binding with a radiolabeled neurotransmitter like norepinephrine or radiolabeled acetylcholine. The affinity of that is 10 to the minus 6. You have 0.01 seconds in order to proceed with that experiment before you get more than 10% loss of your radioligand. So you would never use labeled acetylcholine or labeled norepinephrine to do a radioligand binding experiment because you simply don't have time to deal with it. On the other hand, if you've got that super duper fantastic award-winning antibody that has a 10 to the minus 12 molar affinity, you've got over a day to do this experiment, all right? So you could start it on Friday afternoon and go off to the beach and come back and you'd still be in good shape. So once again, this is not something you're going to have to memorize, something that you have to know, but this is just a really good example of how practical this relationship between affinity and kinetics really is. And I'm going to actually come back to that actually several times. I'll come back to it today, and we'll see it again next week when we talk about G proteins. All right, so that's it for kinetics. Like I said, the only mathematics you really need to know is that relationship between the KD and the dissociation and association rates. That's it. Now, the other assumption that I didn't really talk about in our discussion last time was the fact that I have assumed in all of the equations and mathematics that I gave you that the receptor is a static system. It doesn't change, all right? It has an affinity for a drug, it has an affinity for a hormone or a neurotransmitter, and that affinity stays the same, it doesn't alter. But physiology doesn't work that way, and receptors change as a function to their exposure to agonists. This is a physiological function. It is common to all biological systems. And the process is known as homeostasis, okay? So biological systems tend to try to maintain their own equilibrium, okay? They like to stay where they are, and if they're pushed out of equilibrium, they respond by counteracting in the other direction to restore a homeostatic balance. So that means that if a system is exposed to chronic agonists, that means the receptors are being overstimulated. And if receptors are being overstimulated, they respond by desensitizing or producing a decreased response to the agonist. So this is the most common form of receptor regulation. There's also another form that I'll talk about in a minute, which is called supersensitivity, which is exactly the opposite. But let's focus our attention on desensitization, because this is by far the most common physiological regulatory mechanism for receptors in a whole variety of systems. If you have a receptor in the nervous system that's being stimulated by an increased release of neurotransmitter over a long period of time, it will desensitize. It'll desensitize to compensate for the fact that it's being overstimulated.
So
Yes?
is that like becoming tolerant?
Yes, this is one potential mechanism of tolerance, all right? There are two types of tolerance that you can have for drugs. One is called a metabolic tolerance, and the other is called a pharmacodynamic tolerance. This is pharmacodynamic tolerance because it's produced by a change in the receptor. Metabolic tolerance occurs by metabolism in the liver. So a lot of drugs are metabolized in the liver. It's called the first pass effect. And if the drug it can induce its own synthesis, which a lot of drugs can, you can become tolerant to it because the drug is being metabolized so quickly. So tolerance is part of that. But desensitization doesn't always lead to tolerance. It can lead to a variety of other things. So the mechanism for desensitization is quite complicated, and it depends upon what type of receptor you're looking at. I'll show you an example of that in just a few minutes. So desensitization is a general term, and all it means is a decreased response to the agonist. So if an agonist produced a certain effect acutely, okay, with a single exposure, after chronic exposure, and the time of that exposure can vary depending on the receptor that you're looking at, after chronic exposure, the response to the same concentration of agonist will be diminished. Okay? Desensitization is normally reversible. Okay, sometimes it's very rapidly reversible, sometimes it's slowly reversible. But once you eliminate the chronic exposure to the agonist, under many circumstances, the receptor will switch back to normal again. And I'll show you the molecular mechanisms of that later. But there comes a point at which it becomes pseudo-irreversible, and under those conditions, the receptors undergo what's called downregulation. Downregulation simply means a decreased number of receptors. So initially, you have a desensitization response, which is a reversible, decreased response to the chronic exposure to agonists. But if that chronic exposure continues and continues for a very long period of time, then the cell will respond by metabolizing the receptor and actually sequestering the receptor inside the cell and eliminating it. Okay, and so that the downregulation simply refers to the sequestration and metabolism of the receptors, which is referred to as a decreased number of receptors or a decreased Bmax. Yeah.
So I thought downregulation was like the direct method to desensitize the cell. What is an example of that desensitization?
I'm gonna show you two different mechanisms of desensitization. One, the ionophoric receptors, I'll talk about in just a few minutes, and then next week we'll talk about the G protein mechanisms. So you'll see, particularly when we talk about the G protein receptors, you'll see a very precise mechanism that leads from desensitization into downregulation, and it's very well understood. So let's hold off to that. Okay. Now I call this a pseudo-irreversible response downregulation because it's not completely irreversible. The cell can respond ultimately by synthesizing new receptors, but that might take days or weeks or whatever. All right. So that's desensitization. And here's actually a slide that really addresses your question about tolerance, okay? So desensitization is a loss in receptor sensitivity following chronic agonist exposure. There are a variety of physiological consequences to this. Tolerance, of course, is a long-term loss. Tachyphylaxis is a term that's primarily associated with the autonomic nervous system, which is a short-term loss of transmitter-mediated it really is. It's not a receptor-mediated phenomenon. It's more of a transmitter-mediated phenomenon. And once again, downregulation, which I've talked about already, a decrease, a loss in the Bmax. So this is what it would look like on a receptor binding assay. Suppose you exposed an animal to a chronic, or cells to a chronic agonist, okay, and you let the receptor, let's say, exposed to this for several hours or for several days or whatever, it depends on the system you're looking at, and then you ran scatter plots of radiolabeled drug binding, okay, to determine the effect of a chronic drug on the system. And what you see here, so this is a scatter plot. Remember I told you about this last time. It's bound over free versus bound on the x-axis, and that gives you a straight line. And so here's your control binding. So here is the binding in the cells that have not been treated with agonist, and here is the binding in the cells that have been treated chronically with the agonist. And this is the reason why we do scatter plots, because even an idiot can look at this and see, look, this is very simple. What you've got here is a parallel shift to the left in the line. So there is remember that the Bmax is the x-intercept, and the slope is the KD. Well, the slopes are the same here. So you don't need to calculate, you don't need a computer to see what's going on here, okay? The slopes are the same, so immediately you say there's been no change in the drug affinity for its receptor. What's changed is a decrease in the Bmax, very simply. Now, there are all different kinds of ways that an experiment like this could turn out, but this particular result is a classical result of receptor downregulation, where the cells have responded by decreasing the number of receptors, the Bmax of the receptor. Okay. Now, there are two major classes of receptors that act in the brain, for example, and actually in all kinds of nervous tissue. The ionophoric receptors, which work by changing ionic permeability across the cell membrane, and G protein coupled receptors. So next week I'll talk about G protein coupled receptors. I want to talk a little bit though today about a classical ionophoric receptor, the cholinergic nicotinic receptor. The nicotinic receptor is in the brain, but it's predominantly known as the receptor that mediates muscle contraction. Anytime you have any kind of particularly strided muscle contraction, acetylcholine is released from the presynaptic neuron. The acetylcholine binds to the nicotinic receptor. The nicotinic receptor is an ion channel receptor. So as soon as the acetylcholine binds to the nicotinic receptor, there's an ion channel that opens up, and you get an influx of positive ions into the cell, and that produces muscle contraction. All right. So this is critical for the body. That's why you have things like nerve gases and insecticides that act exactly on the cholinergic system that are so lethal to the organisms that they're exposed to, because this is critical for life. So the nicotinic receptor, once again, is a classic ion channel receptor. It acts very quickly, so it's designed to act within seconds of what's going on. And in fact, it goes on off and on very quickly within seconds. So there's actually, turns out to be 3 affinity states for the nicotinic receptor for acetylcholine. And there's a high affinity state at 1 nanomolar kD. There's an intermediate state at 10 nanomolar, and a low affinity state at 1 micromolar. That's 10 to the minus 6 molar. So in the neuromuscular junction, okay, in the synapse between a cholinergic neuron and the muscle fiber, and of course the muscle fiber is where the nicotinic receptor is located, the resting nicotinic receptor has a low affinity for acetylcholine, 10 to the minus 6 molar. And why is that? Because there's an enormous concentration of acetylcholine that's released by these nerve cells. And in fact, some people have even measured the concentration of acetylcholine in that synapse to be anywhere from 10 micromolar all the way up to 1 millimolar. So, you know, when you get a release of acetylcholine from those nerve cells, you're flooding the synapse, all right? And so normally what happens is that this very high concentration of acetylcholine, the acetylcholine will bind to the nicotinic receptor, and because it has such a low affinity, remember the relationship between affinity and dissociation, the acetylcholine will come off almost immediately. So there's a very rapid influx of cations, then the acetylcholine dissociates, it's gone, it's hydrolyzed by acetylcholinesterase, and the channel closes. So the whole system is designed to open and close very quickly. That's how this whole thing works. Now, so during this activation channel phase, when the channel is open, there is a moderate acetylcholine affinity, and we don't need to worry about that activation phase very much. Now, suppose you overstimulate the system. Now, you can do this in 2 ways. You can stimulate that presynaptic neuron, that cholinergic neuron, so you flood the system with more and more acetylcholine for a long period of time. Or you could simply give the cells or the animal or the patient a cholinergic agonist, a nicotinic agonist for a long period of time. Either one of those will do the same thing. Now remember, this whole system is designed to go very quickly. But now when you flood the system with acetylcholine for a long period of time, the system responds, the nicotinic receptor system responds by becoming desensitized. All right? So the nicotinic receptor basically says, I've had enough of this, I am shutting down, okay, because I am being overstimulated. So how does the nicotinic receptor function to become desensitized? Well, what it does is it switches from a low affinity state into a high affinity state. So it switches from its basal state of 1 micromolar, which is 10 to the minus 6 molar, all the way up to 1 nanomolar, which is 10 to the minus 9 molar. Now, let's go back. Look at what that's going to do to your dissociation rate, all right? We talked about this before. It switches from, okay, 1 second all the way up to 100 seconds. So what's going to happen now in the desensitized state, at that high affinity state, the channel becomes closed, Okay, so no ions can go into the cell. There's no activation of the muscle cell. There's no contraction of the muscle cell. The system is shut down. And now, because acetylcholine is bound at a nanomolar affinity, it dissociates much more slowly. Okay, so that's the whole idea of this. Is by switching to a high affinity state, the acetylcholine stays on there, and as long as this acetylcholine is on there, the channel is closed and it can't open again. Now, eventually, the acetylcholine will come off; it'll dissociate, and once that happens, the receptor can then switch back into its resting state again. But once again, if there's a continued presence of a high concentration of acetylcholine for a long period of time, this receptor will stay in its desensitized state. So here's an example of a receptor that changes its ability to bind to its neurotransmitter by changing its affinity state. And in this particular case, it changes it from low affinity into high affinity. That may sound weird, okay, because you're thinking to yourself, you would think that a high affinity state would be the active state, but in this case it's not. And the reason for that is because this receptor is designed to act quickly. And the whole reason for this is to have a very rapid termination of the acetylcholine effects so that the next round of stimulation can occur. Now, next week we're going to talk about G-protein coupled receptors, which work in a completely different way, and in fact almost virtually opposite from this. Yeah?
If we continue to get cholinergiomimetics for this patient,
I'm sorry?
if we continue to get cholinergiomimetics, will the receptors will be downregulated then?
Yes, if it's stimulated for a very long period of time, the nicotinic receptor can be downregulated. But it would take a long time to do that. This receptor is really predominantly working through this desensitization mechanism like this. So it's not as downregulation is not as common in this system as it would be for the G-protein coupled receptors. This has by the way, this has very significant clinical relevance when you administer a nicotinic cholinergic agonist. Sometimes that's done during surgery to relax muscles, but you've got to be very careful because you could actually produce very quickly a desensitized receptor, and that can be a real problem. So once again, very relevant, but really illustrates this basic relationship between kinetics and affinity. All right, we talked about desensitization. Now let's talk about the opposite. Once again, remember the receptor is designed to change its function depending upon the environment that it's in. So when it's overstimulated, okay, it'll switch back into a desensitized form. Suppose it's understimulated, okay? If that happens, you see supersensitivity. Once again, the receptor trying to restore its homeostatic balance. So in this particular case, the receptors will undergo a supersensitivity phenomenon. Now, supersensitivity is much easier to understand than desensitization. Desensitization involves a lot of different steps. You've seen it already for the nicotinic receptor. Next week, we'll talk about the even more complicated example of desensitization for G-protein coupled receptors. For supersensitivity, it really is very simple. In virtually every case that I'm aware of, supersensitivity involves an upregulation, an increased number of receptors. All right. So basically, if you understimulate the receptors, you get an increased response to the drug, and that's produced by the cell increasing the number of receptors. This is, in fact, a physiological or pharmacological example of spare receptors or receptor reserve. Because what you're doing here, by what the cell is doing by increasing the number of receptors, it is increasing its sensitivity to the drug. We talked about this last time. I showed you how you could actually use the Langmuir equation to calculate, what kind of increased sensitivity you're going to get by upregulation. Now, how do you produce supersensitivity? Basically, two ways to do it. One is to administer an antagonist chronically, all right? So remember, an antagonist is a drug that binds to the receptor and does not produce a biological response. So what you're doing here is by throwing an antagonist in there, you're blocking the endogenous neurotransmitter or hormone actions. And so that cell that contains a receptor is now being understimulated. Now, if you do this for a few minutes or a few seconds or so, that's really not a problem, okay? The antagonist has its effect, it dissociates, and the receptor now becomes, activatable by its agonist. But if you treat chronically with an antagonist, so that the receptor in that cell becomes chronically understimulated, the cell will respond by increasing the synthesis of receptors to try to restore the sensitivity of that receptor back to normal. So that's a classic example of that. The other example is what's called denervation supersensitivity. And this is a phenomenon that occurs in the nervous system. So if you remove the stimulation by the agonist, in this case you've got the normal agonist would be a neurotransmitter. The neurotransmitter comes from the presynaptic neuron that innervates that particular muscle or other type of neuron. If you cut that fiber or you lesion it somehow way or another, then you're going to lose the stimulation that's normally produced by release of that neurotransmitter, and that will also produce a supersensitivity phenomenon. This particular experiment, which I took out of the literature, basically is an example of this denervation phenomenon. So this drug here, which is called 6-hydroxydopamine, is a presynaptic lesion drug. Okay, so what it does is it's taken up within adrenergic neurons. Adrenergic neurons are neurons that release norepinephrine and epinephrine. Okay, so when you add 6-hydroxydopamine into the system, whether they're in cells or in an animal or in brain, the 6-hydroxydopamine is specifically taken up within the nerve terminals of the adrenergic or norepinephrine-containing nerve cells. And in about a week to 10 days, those nerve fibers will be gone. Okay, they will degenerate. They will be lesioned. They won't exist. So what will happen on the postsynaptic neuron, which has adrenergic receptors, in this case these are alpha-1 adrenergic receptors, those adrenergic receptors will become understimulated because there's no longer any norepinephrine for them to bind to. So over a period of about 2 weeks or a month or so, something like that, after administration of the 6-hydroxydopamine, you will begin to see supersensitivity. And this is what that experiment looks like. So if you take brain tissue from the rat, which has not been treated with the 6-hydroxydopamine, here's your scatter plot. And here, these are alpha-1 receptors, okay? So these are adrenergic receptors that are present on the postsynaptic cell, okay? The cells that are normally being innervated by the norepinephrine. So here are the alpha-1 receptors in the animals that have been treated with 6-hydroxydopamine. Once again, here's the advantage of a scatter plot. You don't have to calculate the math here. You can look at this precisely. You can see what's happened here. Once again, there's no change in the slope, so there's no change in the affinity of these alpha-1 adrenergic receptors for drugs, but there is an increase, okay, in the Bmax, an increase in the number of receptors, and that is a classical example of receptor supersensitivity. This is a physiological and clinically relevant phenomenon. Once again, anytime you have a lesion of any kind of pathway in the brain, you'll get this kind of phenomenon happening. Clinically, there's a very well-known response called tardive dyskinesia. Tardive dyskinesia is a physiological response that occurs in schizophrenic patients who are chronically treated with antipsychotics. Antipsychotics are drugs that are antagonists at dopamine D2 receptors, and schizophrenic patients are chronically treated with these drugs over a period of many years. So over a period of chronically, that's over a year or so, they begin to develop very severe motor symptoms. And And that's what tardive dyskinesia is. So if you see a video, for example, of a schizophrenic patient, a severe schizophrenic patient, you'll very often see them exhibiting a lot of very strange motor movements. They'll be jittery and their hands will be going all over the place. They'll have nervous tics in their face. A lot of people think that this is part of schizophrenia. It has nothing to do with schizophrenia. That's the motor problems are the tardive dyskinesia, which is a result of supersensitivity of dopamine receptors after chronic blockade by the drug. So once again, just like tolerance is a great physiological example of desensitization, tardive dyskinesia and other kinds of things are the physiological consequences of supersensitivity. I'm skipping that. Okay, any questions about receptor regulation? Okay, so in this last part of the lecture, I'm going to talk about alternative uses for receptor binding. So what we've talked about so far is the ability to use receptor binding as a screening tool for designing new drugs, but there are other things we can do as well. Drug screening, I'll talk a little bit more about that, distinguishing agonist and antagonist binding, and localizing receptors in the brain. So I talked a little bit about this last time, and that is the fact that drug companies utilize radioligand binding assays to screen for new drugs, to produce produce a library of drugs that they can use to move on to the clinic. And I told you last time that it takes almost 1 million of these compounds in order to really see what's going on, to develop one single compound that goes to the clinic. Here's an example of this. This app just very relevant to our just completed discussion about tardive dyskinesia. Here are a series of drugs that are used to treat schizophrenia, the classical neuroleptics. Okay, so these compounds have been used since the mid-1950s to treat the fundamental symptoms of schizophrenia. Some of them are more potent than others. So on the y-axis here, I'm sorry, the x-axis, we've got a logarithmic plot of the concentration of drugs in blood, in plasma, that are required to produce therapeutic effects in schizophrenics. And on the y-axis, we've got the K, it says let's say the KD, I'd call it the KI, anyway, it doesn't matter. KD or KI of drugs at D2 dopamine receptors, okay? So a very potent, we have a whole series of very potent neuroleptic drugs in vivo, for example, haloperidol, trifluoperazine. These compounds are very, very potent. They're used in the clinic at very low dose levels. Notice that they also have very low KD values, okay? So this is exactly what you would predict. You have weaker drugs like chlorpromazine, for example, and other drugs like sulpiride and others, which are very weak in vivo, also very weak in binding to the dopamine D2 receptor. You also, by the way, parenthetically, have a series of compounds known as the atypical neuroleptics like clozapine that don't bind to the D2 receptor, so they fall off of this line. But if you're a drug company and you want to design new neuroleptics that are acting on dopamine D2 receptors, you use the D2 radioligand binding assay as your initial screen. And you can tell from this, okay, that once you obtain the KD of the drug at the receptor, which you can do in 1 day in a radioligand binding assay, using exactly the techniques that I talked about last time, you can in fact predict how potent it's going to be in vivo, very simply. And not only that, for these drugs, we understand where their side effects occur. So side effects of neuroleptics occur when they're binding to cholinergic receptors, when they're binding to noradrenergic receptors, like alpha receptors. There are a variety of receptors that mediate negative side effects for these drugs. So if you're developing a new drug, all you have to do then is measure their affinity not only at dopamine D2 receptors, but at cholinergic receptors and alpha adrenergic receptors, and that will tell you in basically one day, not only how potent the drug is going to be as in treating schizophrenia, but it will also allow you to predict how many side effects the drug is going to have. So in the old days, pharmaceutical companies had to do a whole variety of behavioral tests in animal models. It would take them a year to really understand what's going on here, which we can basically do in a day or two in radioligand binding. So this ability to screen compounds has really made an enormous difference in our ability to make novel drugs. Okay, so but it does. I'm going to skip this part and go on to this next part. Drugs, of course, can be one of two different pharmacological categories. They're either agonists. Remember that an agonist binds to the receptor and produces a biological response, or they could be an antagonist, which means that they'll bind to the receptor and they produce no response. Radioligand binding can distinguish between agonist and antagonist, and the way that it does that depends upon the type of receptor that you're looking at. This is a particular example of a G protein coupled receptor, and we're going to talk more about the mechanisms of this next week, but I just wanted to introduce this topic today. Guanosine triphosphate is an important component of a G protein coupled receptor. It's part of what makes the receptor really function very well. And guanosine triphosphate, GTP, binds specifically to a component of the receptor which allows its function to occur. If you add in a lot of GTP, you change the state of the receptor from a high affinity state for agonist into a low affinity state. And you can see that here. This is radiolabeled agonist binding, and as you're adding more and more GTP in this thing, you're decreasing the amount of binding. And the reason it's decreasing is because the receptor is shifting from a high affinity state into a low affinity state. But for antagonists, it doesn't matter, okay? Antagonists don't care whether the receptor is in a low affinity state or a high affinity state. They bind equally well. So it doesn't matter how much GTP you put in here, the antagonist binding stays the same. Here is a complicated looking graph which illustrates this phenomenon of how these receptors shift back and forth from high affinity states into low affinity states. So this is a dopamine receptor binding assay. So this is a displacement assay, okay? The unlabeled drug is dopamine itself, all right? And the radioligand is something called tritiated domperidone. Okay, so that's a radiolabeled antagonist. So this is a drug that binds to D2 dopamine receptors. And so this is a classic displacement curve using unlabeled dopamine to displace tritiated drug binding to this D2 dopamine receptor. There's a lot of log units across here. He's going all the way from 10 to the minus 11 to 10 to the minus 2. So he's looking at 9 orders of magnitude of drug concentration. Now remember that I told you before last time that there's an easy way to determine whether or not the hill slope of a particular curve is 1 or less. And that is by counting the log units that it takes for full displacement. So it looks to me like this curve here. Let's ignore this one over to the right now. Let's just take a look at this one. It looks to me like this curve here starts at about 10 to the minus 8th molar, and it looks like it ends at about 10 to the minus 5 molar. So it's taking 1, 2, 3 orders of magnitude for dopamine to displace this curve. So this suggests to me that the hill slope of this is low, and it is. And the reason why it's low is because dopamine is binding to high and low affinity states of the receptor. And I'm going to tell you next week precisely why that is, why this receptor is set up to have 2 affinity states. But here is the high affinity state, and here's the low affinity state, all right? So you've got this mixture of high and low affinity states together. Now, remember I told you a minute ago that if you add in a high concentration of guanosine triphosphate or GTP, you shift all the binding from high affinity to to low affinity. Well, look what's happening here. Look at this curve over here to the right. In this case, this curve has been shifted by GDP, all right? So what's happened here is that all the receptor sites are now present in the low affinity. There are no high affinity sites left anymore. So you've shifted the curve to the right, the new affinity is lower, and look, it's not only is it shifted to the right, but it's also steeper. So if once again, if you count the log units, it goes 10 to the minus 6 down to 10 to the minus 4, okay? That's 2 log units. That's telling you that the hill slope of that curve is 1. And that's because we've only got one set of sites in this preparation, and they're all low affinity. So when you're looking at these curves, people, begin to get trained not only to look at where they are on the graph, but also their general slope. I think this graph would have been a lot clearer if these guys hadn't used 9 orders of magnitude to do the experiment, but I can't. All right, so using radioligand binding, you can distinguish between agonist and antagonist, and you can use these kinds of tricks to determine whether or not the drug you are designing is an agonist or an antagonist. So that's very handy to do. Obviously, for example, if you're trying to design a new schizophrenic drug, all right, a new drug that treats schizophrenia, that's going to be an antagonist at the D2 receptor. So if you find out that your new drug is an agonist, you're wasting your time. You go off, throw it away, and start over again.
Should you know that though from the start? I mean, if you're designing your own drug, you should know if you want to block it or activate the receptor and look at the rest.
Well, you don't know it until you look at it. Now, you can make some predictions based upon what you know about the structure of the drug chemically, but you don't really know it until you actually do the experiment. This is not the only way to do it. There are second messenger assays and functional assays that will tell you this as well. But this is just one way of being able to tell you that. So if you're looking at the very first thing you need to know about a receptor, I told you last time you need to know its affinity for sure and how many receptors there are, but you also have to know whether it's an agonist or antagonist. Otherwise, you're totally up to the last thing I'll talk about is the ability to use radioligand binding to localize receptors in various tissues. I'm using the brain, but you could actually do autoradiography in any tissue, and it has been, particularly PET, and I'll talk about PET in a minute. So when the radioligand binding assay was first developed back in the 1970s, they really began to think of ways to be able to use this to examine where receptors are located in situ, in the tissue itself. Let's think about drugs in the brain. If you know where receptors are located in the brain at a high level of neuroanatomical resolution, you can begin to know the function of that receptor system. Okay? So, for example, dopamine receptors are highly localized in motor systems, and we know that not only by looking at autoradiography, but we also know by the general function of dopamine drugs. When we put a dopamine drug into an animal, we change a lot of locomotor activity. Opiates, for example, like morphine and other kinds of heroin, other kinds of codeine. These are pain drugs, and their receptors are located precisely in those areas of the brain that receive and process pain information. Areas, for example, like the thalamus, for example, periaqueductal gray, the amygdala. These are areas that really light up when you're having a painful experience. And the fact that these receptors are located in this area really shows you how this pain system works. So it's really critical when you're looking at a receptor system to try to figure out exactly where it's located, and that gives you a lot of information about potential function. So how do you do these kinds of experiments technically? The first technique that was used many years ago, and it's still used, although it's much less common, is a technique which is now known as ex vivo autoradiography. And what you did is you took your animal, you don't use humans for this for obvious reasons. You inject the radioligand IV And then, of course, the radioligand, the radiolabeled drug will then go into the brain, bind to the receptors, and once it's bound to the receptors, then you can then sacrifice the animal, remove the brain, and section it for autoradiography. And so you'll, by taking the sections of brain and putting them against film, you can see precisely where the radioligand, the radiolabeled drug was associated. This worked very well, but it was really restricted for a couple of things. First of all, radioligand stability. So if the radiolabeled drug is metabolized at all after it's injected, it's never going to get to the brain, okay? So that's going to be a major problem for a lot of drugs. So you had to choose the right kind of drug that didn't get metabolized before it actually crossed the blood-brain barrier. And that's the second point. You had to have good bioavailability. It had to get across the blood-brain barrier to begin with in order to work. So you could have a very stable drug, you could have a drug that was very potent in binding the receptor, but if it can't cross the blood-brain barrier, it's completely worthless for this kind of technique. So, you know, this was a very limited kind of procedure. After that, people began to work out techniques for what's called in vitro autoradiography. Once again, these are done in animals or autopsy human brains. And this has become the most versatile method of doing autoradiography. In this case, you don't inject the drug in vivo. It's all done in vitro. The brain is actually taken out from the animal. It's frozen, sectioned, not fixed, because if you fix it, which is a good way to look at a variety of other things, but if you fix it, the receptors won't work. The receptors are denatured and they won't bind anything. But you take these frozen sections and you expose them to the radiolabeled drug with using radiolabeled drug binding just the same way we've talked about before. And you wash the sections and you oppose them to film. And then now you've got these nice films that show you exactly where the receptors are located. This is a big advantage over ex vivo because you don't have to worry about the drug being metabolized. You don't have to worry about the drug crossing the blood-brain barrier. Everything is done in vitro, and it's quite easy. The disadvantage is that film is not particularly sensitive to tritium, and many radiolabeled drugs, as we discussed last time, for very good reasons, are labeled with tritium. Tritium is a great drug for labeling drugs, as we talked about. You get a nice high specific radioactivity. It doesn't change the structure of the drug. The problem with tritium is it has an extremely low energy, okay, very, very low energy. And so it takes a long time to develop this film, a minimum of 6 weeks, and not uncommon at all to see 20, 30 weeks of exposure to see what's going on. So if you're a graduate student and you're on a time frame of getting out, this is a slow process of getting done. I actually saw one paper in the literature where the films were exposed for 9 months. So that's a long experiment. And you know, if you find out that you've made a mistake, 9 months later you put the wrong drug in, or you use the wrong concentration, or the wrong kind of film, something stupid, 9 months later you're wasted. So this is a problem. Today, people don't use film very much anymore. They use much more sensitive phosphor imaging plates. So this exposure time has gone way down recently. I think now you're talking about a couple of weeks. So technology has improved quite a bit. So this is what autoradiography looks like. This is for opiate receptors. This is mu opioid receptors. So this is the opioid receptor type that binds to morphine and codeine and heroin, most of the drugs that are used for pain in the clinic. This is kind of a false color image. The higher, the brighter the color, the more binding you get. So red and yellow is high binding, and blue is very low binding. And what you see here is this is the front of the brain. This is the back of the brain. Here is a lot of binding in some of the motor areas. But back here, this is the thalamus, which is a major way station for analgesics. Back here is the brainstem, which contains the paraqueductal gray, which is not only an area that receives pain information, but you've also got other areas here that mediate some of the autonomic effects of opiates, most particularly the respiratory depression that occurs in opiate overdoses. So these kinds of images, once again, very informative about how these receptors work. Now, most of the time, you know, in my lectures, I'm talking about classical, you know, receptors that have agonists and antagonists, like opiate receptors, like acetylcholine receptors, dopamine, norepinephrine. Those are called classical agonist receptors because there is an endogenous agonist, it could be a neurotransmitter or a hormone, that normally activates it, and the activation of that receptor by the neurotransmitter produces its effects. But there are other types of receptors, and here's a classical example of this, transporters. So the brain contains a whole series of high affinity transporters for neurotransmitters. This particular one is for serotonin. Serotonin transporter is probably the #1 drug target for developing novel CNS drugs. And why? Because antidepressants are inhibitors at serotonin reuptake sites. The most commonly used antidepressants today are the SSRIs, the serotonin selective reuptake inhibitors. These are drugs that specifically bind to serotonin transporters and block their ability to reuptake serotonin back up into the cells. So, you know, you can do autoradiographic localization of transporters just the same way as you could do for classical agonist receptors. So here is the serotonin transporter. It is present in a whole variety of areas in the brain. You can see here in the midbrain, there's a whole lot of it in the brainstem. But the distribution in the cortical areas of the brain, the higher brain centers, are the areas that most are particularly relevant for the development of antidepressants. So once again, this whole series of autoradiography type studies allow us to really study where these kinds of drug targets are located. Now the disadvantage to all of these autoradiographic techniques is that it's all done on dead tissue. Okay? So if you want to study receptor localization in the human brain, you either have to use post-mortem tissue or some kind of volunteer. Unlikely you're going to get any volunteers. So this is where PET imaging comes in. PET imaging is positron emission tomography. This happens to be a technique that's done very, very well here. We're in Wake Forest, we're one of the leaders in this particular technique. And the big advantage of this is that the receptor binding here is done in the intact living human brain. The principles are exactly the same, okay? We're using a radiolabeled drug, and we can use it to bind to the receptors, and we can calculate KDs and Bmaxs, all this sort of thing. But the difference is that it's done in a living person. The secret to this is the use of extremely high positron, high energy, positron emitting isotopes that have extremely short half-lives. Two of the most common PET isotopes are carbon-11 and fluorine-18. These have very, very high energies, and you would say to yourself, well, gee, high energy means they're not very safe. You know, you're actually injecting this, and that's how you do it. You actually inject it as an intra-arterial injection into the human. Once again, you have to use a drug that can get into the tissue that you're looking at. So if you're looking in the brain, it has to cross the blood-brain barrier. But there are a lot of PET scans that look at peripheral tissues as well, like liver and heart and other things, muscle. But the bottom line is you're injecting this extremely high energy radioisotope. Is this not unsafe? Well, there's two reasons why it's not particularly unsafe. One is that the energy is so high that it just passes through the body. So when you get damage in the cell by radiation, the damage is caused by the radiation being absorbed by some component in the cell. The biggest thing that you're worried about for radiation damage is DNA, all right? So if the radiation is being emitted, it's taken up by the cell, and if it's going out and it is absorbed by something in the nucleus, DNA, you're going to get the potential for genetic damage. These isotopes are so high energy that they're not absorbed by anything in the cell, okay, even bone. I mean, they just go right straight through it, all right? So if you don't get any absorption in the nucleus or other areas, you know, the potential for damage is relatively low. The other reason why it's not particularly unsafe is that the half-lives are incredibly fast. Remember, we talked about half-lives. Tritium half-life was 12 years. Carbon-14 was 5,000 years. C11 is 20 minutes. Fluorine-18 is 110 minutes. So even if you had the capacity of these things to absorb and produce any tissue damage, they're cleared from the body very, very quickly because of their extremely rapid half-life. Now this is a challenging technique, okay, technically, because you just can't call up your local supplier in Boston and say, you know, send me, you know, C11 raclopride because I want to do some dopamine studies. Because by the time it gets here, there won't be any left. So when you have a PET imaging facility on a campus or in a company, a hospital, you've got to have everything here. You've got to have a cyclotron, okay, which produces the radioactivity. You've got to have a chemistry lab that produces the drug and incorporates the radioactivity. So you got these little chemists, radiochemists, who are creating the isotope out of the cyclotron and then very rapidly making a reaction that attaches that radioisotope onto the drug, and then they run it into the clinic, you know, to inject it into the patient. And all of this has to be done very, very quickly. It's also very expensive. You've got to have a cyclotron. You got to have trained radiochemists. You've got to have nurses. You've got to have, and the scanner is very expensive because you got to have this scanner which can detect the radioactivity and examine what things are. So PET is not something that you use for routine sorts of things, but it is extremely useful for detecting a whole variety of things that you simply can't do by any other methods. For example, there are certain proteins that are highly expressed in early growing tumors. Okay, people can design radiolabeled drugs that bind to these specific proteins, and then you can do a PET scan, and if it shows up in the area that you're looking for, in the liver, the breast, or whatever, you can actually diagnose tumors much earlier than you would by using symptomatic kinds of procedures. So there's a lot of uses for PET. Very interesting. Just not particularly expensive. I mean, not particularly cheap. And here's an example of a PET study. Okay, so let me see if I remember how this works here. So let's skip this 5-HT2. Well, the experiment was done with two different PET tracers with human volunteers. This was done with C11 raclopride, which is an antagonist for D2 receptors. This one was done with C11 N-methylspiperone, which is an antagonist for serotonin 2 receptors. So when you inject the C11 raclopride into a human brain, you look at the localization here. About 90% of that label is localized right in that area. That's the striatal area, the basal ganglia. This is the area that mediates movement, and so we really think of dopamine as a movement sort of area. Now, this particular patient was pre-injected with haloperidol, unlabeled haloperidol, which is a potent D2 agonist. So once again, if you pre-inject with the unlabeled haloperidol is going to bind to those D2 receptors, and so then when you inject the C11 raclopride, you're not going to see as much labeling anymore. This is a classic displacement study. So, I showed you these graphs of displacement that you can do in vitro and in cells, but you can actually do the same kind of displacement study in vivo in a living human. Clozapine, which is a what's called an atypical neuroleptic. Okay, it's a compound that doesn't bind very well to the dopamine D2 receptor. These people were injected with unlabeled clozapine in advance, and you can see that the labeling here has been reduced, but not by nearly as much as haloperidol. Now, the aim of this particular study was to bring forth the idea that clozapine was not was working at 5-HT2 receptors, and so this study on the bottom demonstrates that. Okay, so here are the 5-HT2 receptors. If you inject with haloperidol beforehand, you don't see any decrease in labeling because haloperidol doesn't bind to 5-HT2 receptors. But if you inject with clozapine, you see this dramatic reduction in labeling, once again indicating that the clozapine is acting on the 5-HT2 receptor. So the point of all this is not to try to confuse you with these drug names and so forth, but to really illustrate the kinds of things you can do with PET studies, and they're pretty sophisticated. Okay. All right. Questions on any of that? Yeah.
So for the up and down regulation of receptors, I'm a little confused on one thing. So I'm thinking of like behavioral sensitization to stimulant drugs like cocaine and amphetamine. So if you're increasing your tolerance to the drug, I'm assuming you have a like down regulation or a desensitized response, so you have to increase and you get like tolerant to it. But then during the abstinent period, and like with the stimulant drugs, you have normally like an increased response to the drug when you take it again, and you have that behavioral sensitization. So then would that be the opposite? You have a super sensitivity and like an upregulation of the receptors? Is it like both that happens?
You picked the most confusing example of chronic drug effects that you could possibly imagine, all right? Because behavioral sensitization to psychostimulants is exactly the opposite from what you would predict, okay? Because you're chronically administering a psychostimulant which increases dopamine, okay? So it should be desensitizing all those systems, but you're getting a behavioral supersensitivity, okay? So the easiest way to address your question is we don't know what causes behavioral sensitization with psychostimulants, but there are a number of theories for it, and it's the psychostimulants are the only example that I know of where an agonist-like drug produces a sensitivity. It's more of a behavioral effect than it is a pharmacodynamic effect. Part of what's happening is drugs like cocaine and amphetamine act very quickly, have very rapid pharmacodynamics, and they're extremely rewarding. So animals get behaviorally sensitized to them because of the reward pathways that are involved, and so there's part of that involved. But we could probably spend an hour talking about that. In fact, you should do that in a journal club sometime. It's very, very.
So it's not necessarily related to.
It's not behavioral sensitivity. Behavioral sensitization by chronic psychostimulants is not related to either receptor desensitization or receptor supersensitivity. It's something else.
Thanks.
All right. So I want to spend the last 5 minutes. What is it? Alt-tab, you said?
Before you do that, I have a couple announcements.
Okay.
The test is on Friday at 9 a.m. in this room. Graph paper will be provided. Oh, you want to take it from here?
Yeah, I think from here. Hey everyone. So Friday, test starts at 9:30. So 9:30 to 11 is the regular time. And it's going to be in this room. And we provide you with a graph paper if you need to draw something. At the same time, there's no calculators or cell phones allowed. And if you have questions, ask us before the exam. And also, there's a question regarding homework assignments. We really did not receive all homework assignments from everyone for the past 3 assignments that were due. I think the last one was due yesterday. So there's delivered and executing assignments and the check